Hi everyone, Mrs. Below here. In this video, I'm going to be going over the light reactions and the dark reactions, which are two big focal points of the photosynthesis unit. So first, I'm going to start with the light reactions. And note that this is occurring in the thylakoid. So in a chloroplast, it has a double membrane, double layer, and then it has these little stacks. Those little stacks are called grana or granum, depending if it's singular or plural. And one of those individual discs, that is a thylakoid. And so this is a very zoomed in picture of one of those disc, pancake looking things, which we call the thylakoid. And that's where the light reactions occur. So I'm going to work my way left to right in this diagram. Note that when this is actually happening in a cell, in the chloroplast, in a plant cell, it is all simultaneously happening. So everything's happening at the same time, but it's easier when we're talking about this just to go in order, left to right. So we're actually going to start with photosystem 2. And this one is photosystem 1. They're named for the order in which they were discovered, not the order in which they occur. Now, at both photosystems, you're going to notice there's these green dots. So note that those green dots represent chlorophyll. And chlorophyll is the pigment that gives plants their green color. And it's also the thing that will absorb sunlight. So you can see here that that light energy will get absorbed by the photosystem, specifically the chlorophyll that's in that photosystem. And it looks green because green is the only color that plants cannot absorb, and therefore it is reflected. So plants are absorbing the other colors like red, blue, violet, orange, etc., and they reflect green. So we know that plants need sunlight, and plants also need water or H2O. And we're going to see H2O being split at photosystem 2. So it will be split up into its individual elements. So we will get oxygen, which is our byproduct or waste product. This is how plants make the oxygen that we as, humans and other animals breathe. We will get the H+, and you can see a bunch of those over here, which we'll talk about later. And then we also get the electron. So the electron will enter photosystem 2. And along with that sunlight, it's going to energize that electron. So now that electron has energy, so it is ready for the next step. And the next step is going to be the ETC, the electron transport chain. So now that this electron has energy, it's going to move through the electron transport chain. So remember, the electron transport chain is powered by these electrons, which came from the splitting of the water and were energized by sunlight energy from the sun, and that was absorbed by chlorophyll in that photosystem, photosystem 2. And so these electrons power the ETC, so it can act as an active pump. So it can actively pump H+ across the membrane, and this is a type of active transport. We are just using the energized electrons to power this. Then, once those electrons have done their job at the ETC, they will enter photosystem 1, where they will again be re-energized by energy from the sun, which is being absorbed by chlorophyll. And instead of doing like another ETC with that active pump, they are going to be picked up by NADPH plus or NADP plus and an H plus. We combine those three things together to form NADPH, which is one of the things we were trying to make at the end of this reaction to use in the next set of reactions. So you actively pumped all of this H plus into this space. Well, now it has to go somewhere. So those H pluses will leave through this purple thing right here, which is both a protein channel and an enzyme, and that is ATP synthase. And remember that process of those H pluses moving passively from high to low concentration, that is a type of passive transport called chemiosmosis. And remember, that is a type of passive transport. So that just happens without energy being required. And as this is happening, that powers ATP synthase so it can make ATP. Another thing that we needed to make at the end of these reactions. And both of these things will now go to the dark reactions. We can abbreviate it RXNS / Calvin cycle. So in this step, we were inside of the thylakoid, that little pancake-like thingy. In the chloroplast, we used sunlight and water. We got oxygen out of this, our byproduct or waste product, and NADPH and ATP, which we're now going to use here in the dark reactions, also known as the Calvin cycle. And this is happening in the stroma, which is that space. So I'll point to it up here, which is that space that fills the chloroplast. So it makes sense. So this space here in this diagram, the stuff that fills the chloroplast outside of the thylakoid. And the dark reactions, or the Calvin cycle, we're going to break down into 3 main phases or steps. So the first phase, phase 1, is what we call carbon fixing or carbon fixation. And that is when we're going to see the role of carbon dioxide, which is CO2. We are going to combine that with this molecule here, and this molecule is RUBP, which stands for ribulose biphosphate. So we're going to combine those two together to make this smaller 3-carbon, a bunch of these small 3-carbon molecules. In the second step, we call this rearrangement. Technically, it's called reduction, but just because we don't really get in depth with chemistry in this class, reduction sounds like you're losing things, when in chemistry it means something else. So we're just going to call this rearrangement. It's just chemical reactions. So now we are going to use that ATP and NADPH from the light reactions. The ATP will give us a phosphate. So then it becomes ADP because it lost a phosphate. And then the NADPH will give us a hydrogen. So then it goes back to NADP plus. These both go back to the light reactions to be reformed. And then I know these two look the same. Technically, chemically, they are different. And then we get a bunch more smaller 3-carbon molecules. And then in the third phase, we call this regeneration. Okay. We are going to regenerate RUBP. So you can see here, those molecules continue, and we have to use some more ATP to regenerate that RUBP. So I'm going to say using some more ATP here. And again, that becomes ADP and goes back to the light reactions. But then also, we are going to set aside a molecule. So we're going to set one of these molecules aside, and that is so that we can make glucose, which is the goal of the goal of photosynthesis is to make glucose, which is C6H12O6. We could use it to make other organic compounds depending on what the plant needs. But the plant can use this as a source of energy. So it can do cellular respiration, make ATP, or you can eat the plant and you get the glucose. Now, this molecule of glucose will be formed after you have gone around that cycle 2 times. Once you've gone around twice, then you will make 1 molecule of glucose. So if you had to do some math in terms of numbers, you could look at this and say, oh, there's 6 NADP plus that are used in 1 go around. So after 2, that would be a total of 12 NADPH that would be used.